This tutorial for the Integrated Microbial Genomes Annotation Collaboration Toolkit will focus on the sequence-based similarity module, and in fact we'll focus on one of the 4 items in the sequence-based similarity module, the first item called BLAST. So as with any beginning to this, I need to log in to my student account. And now I will select the course that I am going to be enrolled in. I will select my assignment. And in my assignment here, I will open up in a new tab, the Gene Details page, just to have it around. And of course, a new tab, my lab notebook link, where you can see the sequence-based similarity module. And finally, I will go to the lab report page just to show you that I'm going to be gathering the information for the sequence-based similarity part of my lab report, but we have some other. I'm just going to close that down now so I can stay focused on what's going to happen here. So back to the sequence-based similarity module. We're going to use some bioinformatic tools now to investigate the possible roles and functions of the machine annotated gene that has been assigned to you as a student to examine. As been described before, we often think of this genomic database as a museum, and the museum contains a large number of DNA sequences, and we can compare our DNA sequences with DNA sequences in other museums, sort of akin to comparative anatomy, wherein you might have an unknown bone and you bring your unknown bone to the bone museum and show it to a curator, and the curator can help you identify the bone and classify the bone and make amazing deductions about the role of that bone. So in this particular tutorial, we're going to start off with BLAST. BLAST is Basic Local Alignment Search Tool, and it is the gold standard to begin our comparison of our DNA sequence against the database of DNA sequences. Before we can go on, though, we need to go back, and we are going to, in fact, not do DNA BLAST in this exercise. We're going to do protein BLAST. And so we need to go back and grab our protein sequence with a copy and paste. We need to select the protein BLAST right here and paste our FASTA sequence into this box and push BLAST. Now you're going to see something that takes 1 to 2 seconds. This is an example of the amazing power, supercomputing power, because we are, in a sense, nearly done with this. Well, I maybe have spoke too soon. 7 seconds now. While it is continuing to search, I will go back and remind you that this is BLAST, Basic Local Alignment Search Tool. This is a tool. This comparison tool is something that is done every day by researchers in the biomedical science fields and biotechnology. Here is the result of that BLAST. What you're seeing is a color key, the score. We're getting a score, and that's what I'm really going to talk about at this point. We're getting a lot of really good hits, and this very, very small, so I'm going to blow this up a little bit here. Note that the top hit in this search is the query. That is, we searched a database and we found ourselves. So this, in fact, is not a hit. It is a validation, sort of an internal positive control that, in fact, the search is working. What we will do is we'll select the second and third hits and use these to fill up our lab notebook in the sequence-based similarity module. So we're going to go through BLAST, and we're going to line this up and say, What is it that I need to put in my lab notebook? And it says I need the gene product name of my top hit. I need the organism of my top hit. I need the alignment length of my top hit, the score, and the e-value, as well as the alignment. And I have to do this with the second hit as well. So that's as far as we're going to go in this tutorial, and I'm going to do a lot of copying and pasting to get this in there and get this idea out there. So the gene product name of my top hit. Yeah, well, we can just click here, and I go right down to that hit. And that gene product name is a molybdenate ABC transporter ATPase subunit. I'm going to copy that, and I'm going to paste it in right there. Then it's asking me for the organism, and this is Thioalcamicrobium aerophilium. And so we're going to take that, and we're going to place it right here into the organism, and then we're going to try to figure out what is the length of that alignment. So when we look up here, how long was that alignment? So here we have our query that begins at amino acid 1 here, and it's a 358 amino acid protein, and so here's the 358 amino acids. Now we're looking at the subject. It begins at amino acid 1, and this is unusual. In fact, it ends at amino acid 358. So the alignment length here, as it's described, is 358. I'm not going to copy that. I'm just going to paste it in. So now we have the two parts of BLAST that really are the learning objectives and the learning outcomes that are associated with understanding sequence-based similarity searching. And that would be right here, something called a score, measured in bits, and something called an expect value. So as I'm copying and pasting these into my lab notebook, I'm going to discuss the role of the score and the e-value in your interpretations. We'll go over here and grab the score of 455 bits. And we'll pop that score right in here. And then grab the e-value, and we'll copy the e-value and paste it right in here. All right. In order to get context into this, the expect value is, in essence, a number greater than 0, but must be less than 1 to be legitimate. The smaller the number, the better, let's say. And that small number represents the chance that this alignment, or the probability that this alignment occurred by chance. So while these amino acids don't line up very well, right here you can see there's a strong alignments along the whole length of this particular protein. The score is calculated by adding a number for each position. So here, when our alignment length is 358, we're able to have 358 opportunities to have a number, a score that aligns and adds up. It's additive. So here M and M line up, and so there's some number associated with that, maybe a 2, for example. N and A do not align, so that's a 0. K and K align, so that might be a 3. So up to this point you have 5. And as we move along, this computer calculates the BLAST score based upon a scoring matrix, which is something that is a little bit farther along than we want to go. So I guess I just want to get the rest of this information so we can finish the BLAST portion of the sequence-based similarity module. And I need to grab, the last thing I need to grab is, wrong one, sorry. I've already, I need to grab that and put my alignment right here. I could, in fact, fix this a little bit by selecting a Courier font. A Courier is a constant width font, and you see that. We might just bold that too so it looks a little bit better in my lab notebook, because I'm an anal retentive in my lab notebook. So I'm going to grab this second hit. So gene product name of the second hit. I'm going to take that guy, pop it in. I'm going to go get my organism name. I'm going to copy this in. We're going to go get the alignment length. That's 369 in this particular case. And I'm going to grab my score. The score is, see, the alignment length is longer, but the score is lower. Put the score in right there. And I need to get my E value, so I can whiz over here and grab this E value. And then grab my alignment length. My alignment goes right here. You can see this alignment is a bit different. It doesn't start in query number 1, but it starts, oh, see, I didn't have it copied there, so I'll go back. And again, you can see how kind of not pretty that is, so we're going to use the Courier font family. And I don't know, let's just experiment a little bit and see. Oh yeah, that looks so much better. I might not even, I need to bold that. I'm going to save my work, as you noticed before. All right, so back to the BLAST page. Quick review: the alignment length, the score, the expect value, the gene name, the organism name. And one last little tip, there are instructions right here you can open up in a new page if this isn't good enough for you. This has been the BLAST region of the sequence-based similarity module.